In both median string problem and the motif finding problem, we have to examine a large number of alternatives, and from these alternatives then we select the best one. Best one means the best possible sequence. For example, in case of motif finding problem, we have to consider all of these alternatives, and from these alternatives we will select the best possible answer, and we will select from the DNA sequences that will be starting from the position S. So we will apply this formula we have seen earlier, n minus 1 plus 1 raised to power t. So by applying this formula we will come up with this arrangement of the arrays. So here we can see we have to calculate the different possibilities within the strings and in the motif finding problem also. So for example, we can see here we have to calculate all the possibilities up till n minus 1 plus 1. So here we can see this 1, 1, 1 and 1 is identical. Again the 3 1s are identical, 2 is different. In the third possibility 3 is different, and we have to calculate up to the level of this. So we have to calculate up to the last string and the array. Second, we will again continue to calculate, and we will see here the number is not different, but here we can see apart from 1, 2 is written here, and again the last element is different, and we will calculate until we will reach to the end, and so on. We will continue to calculate until we will reach up to the last level. So we have calculated all the columns, and we can see here the number is changing, and we will continue this formulating by using this formula until we will reach to the end of the problem, and we will obtain the maximum number of possibilities. From out of these maximum number of possibilities, we will then get the number of the different types of strings. We will see in the next slide. So if we consider the DNA sequences apart from the integer numbers, we can see if we apply that formula n minus 1 plus 1 t, and we use this on the DNA sequences, and we have to consider or L 4 raised to power L. 4 are 4 nucleotides, and L is the length of the mer, L mers. It could be any number of the size of the DNA sequence. So we can see here the same arrangement will be applied on DNA sequence. For example, in the last column we can see A, T, G and C, they are different. All others are same. So we will continue till the second and third and fourth will be different, and we will continue until we will get all the different arrangements of the strings. So by using this formula we will come up with the solution of the all possibilities of 4 raised to power L. So here we can see in the figure number 3, we can see all the different possibilities of 4 mers. It means that the number of string is 4 and the number of alphabets it's 2. So we can use 1 and 2 up to the level of 4 mers, and we will get the possibilities, different possibilities of these 4 mer arrangements of 1 and 2. So by using this arrangement, if we assign 1 number for adenine, 2 for thymine, 3 for guanine and 4 for cytosine, then we will come up with these types of number. Here we can see very clearly that we have replaced adenine, thymine, guanine, cytosine with 1, 2, 3, 4 numbers. So instead of A, we write 1, 1, 1, 1, means that there are 4 A's here. So if it 1, 1, 1, 2 means A, and 2 stands for T. So apart from T is 2 written here. So for by using the same formula n minus L plus 1, we can calculate all the possibilities within a string. So we can, in general, we can consider KL mers in a K letter alphabets. For the motif finding problem, K is n minus L plus 1, but for the median string problem, K is equal to 4 nucleotides. So we can see in the last figure that we have calculated all 2 raised to power 4 mers in 2 alphabets. Here we can see we have calculated the 4 mers in the 2 alphabets. So by using the same formula, we have calculated the different possibilities for DNA strings. We have seen that we can use a tree search to find the various possibilities within the string, and by using that technique we can use the various types of DNA strings within the specific sequences of nucleotides.